So it is my pleasure to introduce our speaker, Baba Otu. In 2008, Baba started her undergraduate at the University of Science and Technology in Ghana, specialized in biomedical engineering. When she was there, she had worked on developing an algorithm to filter arrhythmia problem with the rates or rhythm of the heartbeats in the application of automated external defibrillators. After Baba received a Bachelor of Science degree in 2012, she worked there at the same university as a teaching and research assistant for about 2 years. Then in 2014, she came here to the University of Calgary to start doing master's course under the supervision of Dr. Leping Lee in mechanical engineering. During her master's, Baba worked on how the knee joint loading affects site-specific cartilage gene expression using a porcine model. She received a master's degree in 2016 and decided to do more research, so she started a PhD course in 2017 working with Dr. Walter Herzog. She's been working on dynamic properties of chondrocyte in cartilage, which is related to today's topic. Her work has been getting attention and acknowledged by Answer Create funding and International Society of Biomechanics as well, where she won David Young Investigator Award in 2019. Aside from her academic career, she mentions that she really enjoys organizing social events, including fun trips and parties. And I actually personally know this because when we had a year and the party every year, Baba was always there as a moderator, coming up with the new creative fun games. So if anyone needs to come up with new games, I think Baba is the first person that you want to talk to. Today, Baba is giving a talk entitled as Real-Time Chondrocyte Volumetric Strain Changes During the Unloading Phase of Dynamic Loading. All yours.
Thank you, Sungwon. And I'm just going to share my screen. Can you all see my screen okay? Awesome. Yep. Okay. Before I proceed, let me drag our faces to the corner as well. Okay. Okay, great. So once again, thank you, Sungwon, and thank you everyone for making the time for the seminar today. As Sungwon mentioned, I will be talking about some aspects of my PhD work, and the title is Real-Time Chondrocyte Volumetric Strain Changes During the Unloading Phase of Dynamic Loading. So articular cartilage, as we all know, is a very highly specialized connective tissue that covers the ends of bones of load-bearing joints. And it is responsible for load transmission, as well as providing a smooth lubricating surface to allow low friction joint movement. Cartilage structure and function can be altered during injury or on the onset of diseases such as osteoarthritis. Now, taking a closer look at the tissue, articular cartilage in the knee joint is avascular and aneural. And chondrocytes, which are the only cell type in the tissue, are known to be responsible for regulating and modifying the extracellular matrix in response to changes during knee joint loading. So based on the loading environment, cells can begin either an anabolic or a catabolic process in a bid to maintain the homeostasis in the tissue. The cartilage matrix also is primarily made up of type 2 collagen, proteoglycans such as aggrecan, and a lot of water. So structurally, cartilage is made up of the 3 zones, which is the superficial, the middle, and the deep zones. And these zones are known to have very different mechanical properties due to the differences in the fiber orientation of the collagen, as well as the differences in the shapes of the cells in the zones. There are also differences in the proteoglycan density as we move going to the increasing density from the surface towards the deep zone of the tissue. Because of these differences, chondrocyte deformations when tissue is loaded are also known to be depth dependent. With respect to the mechanics, cartilage can be described as a biphasic tissue with a fluid phase and a solid phase. The fluid phase is primarily made up of water, as mentioned earlier, and this water contains a small amount of electrolytes. And the solid phase is made up of the collagen and the proteoglycans, which we typically refer to as the matrix. So these proteoglycan aggregates, in combination with a fluid phase, provide the compressive resilience we observe in cartilage through negative electrostatic repulsion forces. The negatively charged proteoglycans bind to the water, thereby producing a swelling pressure in the tissue. And this pressure influences not only the hydration of the tissue, but its mechanical response to loading and deformation. So during loading, both the cells and the tissue deform, although differently, because the modulus of elasticity of the cells, that's the chondrocytes, are known to be 3 orders of magnitude lower than that of the matrix. So you might want to think that with the cells being softer, the tissue might kind of behave like a Swiss cheese, with the cells being the holes and doing what the matrix does. We, however, do not expect this behavior also because the tissue matrix is known to be passive, whereas the cells are also known to be active and can therefore actively regulate their volume in response to loading. But really, it is a complex interaction between the cell and the matrix, because if we assume the cell is very soft and there was sort of a fixed connection between the extracellular matrix and the cell, then of course you expect the cell to deform exactly like the local matrix deforms, but that's not really the case. In figure 3, we see a cell that looks perfectly round and symmetric with the green pericellular matrix around it, which is not exactly a correct representation, as has been observed recently by Nkwanmu, Scott Sybil, and others. They found the PCM has very different properties than the ECM, and also the PCM tends to be asymmetric, and that makes the loading very interesting to obtain. Here also, it almost appears that the chondrocyte is continuously attached to the PCM, but that is not also correct, as very direct focal adhesions of the cell to the PCM have been identified as well. And understanding this might not be as simple as this picture might be painting for us right now. Also, when a joint is loaded, the forces are transmitted from the matrix to the cell surface through proteins like integrins. And the mechanoreceptors on the surface of the cell recognize these physical forces and transduce them into some intracellular responses. The cartilage tissue then adapts by remodeling to the new mechanical environment. So multiple signal cascades are regulated by the mechanical load, but ultimately all contribute to the maintenance of the cartilage homeostasis. So cartilage responds to loading both mechanically and biochemically. And mechanical stimulation is therefore an important regulator of chondrocyte metabolism that is required for maintaining the normal cartilage matrix properties. So specifically also, cell shape and volume changes have been shown to initiate the influx of biological processes such as calcium, which might be part of the signaling pathway. So one of the earliest work which gave us insight into chondrocyte morphology changes during static compression was Gerlach's work in 1995. And by using confocal microscopy, cells were imaged prior to compression and after the compression. And at a 15% surface-to-surface equilibrium strain in the tissue, a similar magnitude of local tissue strain was observed in the middle and deep zones, whereas the superficial zone experienced local strains of 19%. With respect to the cell volume, he also observed decreases of 22%, 16%, and 17% in the respective zones. So local strain measurements also in this study were obtained by measuring the changes in the intercellular spacing using the edges of the cells. Another method for measuring local tissue strains used by the same group and others have been by using the nucleus of the cells as centroids. And both of these approaches have been questioned, as we cannot just overlook the effects of the cell deformation on the measurements between the edges. Also, there has been some evidence of nuclear deformations as well during loading. Now, in the area of dynamic loading of cartilage tissue, in this paper by Han, he adopted a clever approach to investigate the viscoelastic response of the cartilage cells, the chondrocytes, in their native environment under both static and cyclic mechanical compression. And they found our cartilage matrix and chondrocyte recovery in situ following mechanical compression was highly viscoelastic. This study focused on the recovery phase of the tissue, the local matrix, and the cell after applying the compression. But however, initially the plan was to investigate the deformations during the cyclic loading. And they found out it was technically difficult to capture the 3D matrix and cell morphology during dynamic phases when deformation occurred rapidly. So based on these discussions, some of the limitations that were identified include being able to only image cells at steady state during static loading and at the end of loading for dynamic cases. And this has been as a result of imaging technique restrictions with a lack of sufficient temporal resolution to record the transient changes in the cell shape. So microscopy has pretty much been a bit slow in scanning the entire volume of the cells, as we could not do this in milliseconds. Also, some test setups only allowed the superficial zone to be imaged. So the objective of the current study is to measure the instantaneous chondrocyte shape and volume changes during the unloading phase of cyclic loading. And from previous work, we observed that cells lose volume both after static and dynamic loading. However, volume loss during the dynamic loading was less. So with respect to the cycle by cycle behavior, we hypothesized that the cells exposed to cyclic loading conditions would deform primarily in the first loading cycles, with little further deformation in the subsequent loading cycles. This hypothesis was arrived at after putting into consideration fluid loss influencing the cell volume reduction with increased tissue compression. So we obtained our samples from the femoral group of skeletally immature pig knee joints, and 6mm diameter plugs are stained for live cells. And for this study, cells in the superficial and middle zones are analyzed. So the experimental setup is made up of a compression device placed on the stage of the multi-photon microscope. And this has an XYZ linear stage adjuster. And next we see the actuator connected, which is then connected to the load cell, which is yellow in the video. Then we have the red connecting rod leading into the bath. So inside this bath, we have a sample holder attached, and inside the sample holder, we fix the semi-cylindrical plug of the cartilage attached to part of the bone. The indenter is also fixed on the connecting rod, and we can also see that it is sealed with a latex to hold the phosphate buffer solution, and the blue LVDT is attached to dictate the displacement signals to the device. So as mentioned earlier, the tissue is stained for live cells, and each semi-cylindrical specimen is oriented such that the compression will be applied along the axial direction. The sample orientation allows for imaging of the cells throughout the depth of the tissue. So in the current study, the superficial zone of the tissue is defined as the 100 micron thickness from the surface of the tissue after we omit the top 20 microns because the cells from those top 20 microns were poorly segmented when we looked at cells from those layers. Also, the middle zone cells were obtained from between 300 microns and 500 microns of the tissue thickness. So for the loading protocol, the tissue is loaded to a strain amplitude of 20% at a frequency of 0.2 Hertz for 100 cycles, which lasts approximately 8 minutes. And prior to loading, stack images of the target cells are collected to serve as a reference. And the subsequent images are collected after cycles 1, 3, 5, 7, 10, 20, 50, 80, and 100. The images are collected more frequently during the first 10 cycles, as we hypothesize that the cells will deform initially during the first few cycles, with little or further deformation subsequently. So the target cells are segmented from the 3D image stacks collected from the microscope. And using the open source package Python Analyst, which was developed by Scott Sobel in our group, the software generates 3D polygonal surfaces for all cells in the reference and the deformed configurations. And then subsequently, the transformation that best describes the change between the corresponding cell configurations are calculated using numerical optimization. So here we see a raw image, and then we see the segmented image, and then we have a combination of the two images to show how the segmented images fit with the raw image. So now looking at the results from the segmented cells of the superficial zone. First, we see the cell in the reference configuration, and when the cell is loaded, we see it go through the different cycles, as can be seen. And as the load is applied, we see the deformation, and going to, we see the deformation become kind of like a bit more less. And here is a second example, also in the superficial zone. And again, we observe the deformation as the load is applied through the different cycles. In the next example, we would observe the cells in the middle zone. And as can be seen, we don't see much of the displacement of the cells as the load is applied and going through the different cycles as well. So now for the summary results, we have the volumetric strain on the vertical axis as a function of the loading cycles from cycle 1 in blue to cycle 100 in gray. There is a lot of results to look at here, but I would like to pay attention to 3 key points which were very consistent in the study. So in the superficial zone here in cycles 3 and 5, we get a relatively big volumetric strain. And the second thing is, as we can see from cycles 10 or 20, there is very little deformation of the cell. And the third result I would like to observe also is that after the initial big deformation, the deformation volume loss we record earlier, not only do we not have a big volume changes from cycles 20 to 100, but also the cell somehow went back to its original configuration, as can be observed. In the case of the middle zone, we observe pretty much no deformation across the 100 cycle loading protocol. So we also looked at the cell deformations in the cell height, width, and depth. And in the case of the superficial zone cells, the highest strain were observed in the cell height, which is the direction of the load. With respect to the behavior of the different cycles, it followed so as I mentioned, the biggest deformation was with the cell height. And as was in the case of the volumetric strains, it pretty much follows the same trend as well. And another interesting thing to observe here is that with the narrowing of the cell height, we would have expected that there might be, like an expansion of the cell width, as has been observed in the static behavior, but the cells in the dynamic loading seem to behave very differently. Again, with the middle zone, we don't see much happening. Also with the cell dimension changes going across the 100 cycles. So as has been seen from the results, the cells were temporarily deformed and they seem perturbed in the first few cycles, lose volume as expected, but also between cycles 10 and 100, they do not seem to change volume anymore. The cells make a recovery to their original volume, which they kind of seem to like. And so the question then would be, why do we not see a similar reaction in the static case, as we saw earlier with the Gillard results, which we have also confirmed with some tests in this project. The cells in the static state deform and continue to remain deformed at steady state. There appears to not only be a difference in the mechanics, but also a very different active volume regulation of the cell. But during the cyclic or dynamic loading, it prefers to return to its original volume and doesn't want to be compressed and retain and release the strain with every cycle as the loading is taking place. It is also known that cells do not only lose or gain volume through mechanical loading, but also by using, as mentioned, clever mechanisms such as the active cell volume regulation to protect itself. And this probably helps it to avoid fluctuations in volume, which may lead to fatigue failure of the cell membrane. So activation of the volume regulatory channels, such as the sodium, potassium, chlorine channels, definitely would play a role in regulating the volume. So the important question one may ask after seeing these results is also, why do we not record deformation in the first loading cycle? And as can be seen from this video, when the tissue is loaded, the cell is displaced first, but we observe bigger displacements with the subsequent cycles. Yeah, so I guess it's pretty much when you load the cell in the first time, I will assume it builds an enormous amount of pressure, and there really will not be that much local deformation as much of the force is taken up by the fluid phase, like if you're thinking of the tissue as the biphasic model. And as we go on with the cyclic loading, we have fluid flow out of the pressure area, and we have kind of like the shift towards the stress being taken up by the matrix and less by the fluid. So that might be the difference between the first loading cycle, where we have a buildup of that high hydrostatic pressure, as mentioned, and with the second, third, and fourth cycles, when the fluid flow has started to happen and things move more, that's when we might see the compression and then the change in the volume. But essentially, the difference with the recovery in the dynamic state as compared to the no recovery that is observed in the static state is something that we cannot really explain at the moment. So however, for future work, we will be looking not only at the cell deformation, but also the local tissue strains. And we think that potentially that might help explain the different behavior we see in the static and the dynamic conditions. And the way we do this would be by using the grid line technology developed by Nkwenmu and Scott Sibyl in our group. And with this technology, we photo bleach the three-dimensional grid into the tissue. And so we have the 3D grid in the neighborhood of the cells. And by analyzing the 3D tissue tensor, we can have a good resolution of the matrix strains and relate that to the cells. These photo bleach, the grid lines, has been shown to not affect the mechanical properties of the tissue. So in summary, we have dynamic deformation of the cell for the first time in a systematic and a controlled manner. And the big result for us is that the dynamic behavior of the cells is completely different from what is observed in the static case. However, most mechanobiological information has been based on the many papers available on static deformation of cells. And we think that the dynamic behavior is infinitely more important because we have shown previously in our group that calcium signaling is directly associated with the dynamic deformation of cells. Other people have shown also that the dynamic deformation and stretch of the cell membrane is important and not the static behavior. So I would like to thank all the following people and essentially the HESA group as a whole for all the help I received with this project. And also thank you to our sponsors. So I would be ready to take any questions.
Thank you, Baba, for the interesting talk. So if I would like to friendly suggest everyone to turn on your camera if you feel comfortable with it so that we can see each other and then have more active discussion period. Thank you for doing that. So if you have any questions for our speaker, please use the icon raise hand button that is found if you go click reaction icon at the bottom of your Zoom screen. If you also cannot find the button, that's still okay. Please feel free to send me a message via Zoom chat, then I'll call on you when it's your turn. Now the floor is open for the questions. Yes, Brent, please go ahead.
Thanks, Baba. One thing I am having trouble understanding is how the cell compressed in every single direction with the applied load. So when you showed the figure where it compressed about its width and its length and its depth, I don't understand how a material would behave that way. Do you have an explanation? Kind of, does it have everything to do with what's going on with the surrounding ECM? I mean, I guess it has to. Were you surprised by that observation?
Yes, I actually was very much surprised, and there was one result that made me go back to look again at all the segmentation. But really, I don't have an explanation at the moment. But yeah, things seem to be quite a bit different with the cyclic loading and tracking these cells. Yeah.
Oh, okay. Is that something that you wouldn't see with just a monotonic loading scenario?
Well, I don't think I have observed that with the previous publications, because typically when you have the compression, you sort of observe a bit of an expansion, right, in the other direction. But yeah, hopefully we understand some more as we continue with the experiments. But at the moment, I don't have an explanation for that.
Okay, and then the other thing that I found really surprising was the fact that the cell would sort of, it would rigidly translate, and at the same time it would start to lose a lot of volume in the first few cycles. And then I guess I'm surprised that it actually is regaining its original shape in the subsequent cycles. I have no idea how that's happening either. It would kind of make sense to me that it would take a while for the ECM to get back to its sort of original shape after you, or while you're dynamically loading it. So how is it, what's happening there? How is it that the cell is then able to recover its original shape while it's continuously being dynamically loaded?
Yeah, I think those are very important questions. And as I mentioned in the introduction, right, I think we've typically, thought a lot about the cells as being attached rigidly to the PCM or the ECM, kind of glued all around. But if we really understand how not just the focal adhesions, but what the interaction is between the PCM and the cell, hopefully we can make much more sense of what's happening. But yeah, I don't really have a good explanation for that at this moment. Yeah.
Yeah, okay. Thanks.
I see Inzu, you have a question for Baba?
Yes, yeah, very interesting topic. Thank you for the presentation. Along with the brand, the question, I think in your microscope setup, you have constraints on all the sides, on all four sides of your sample plug with because your sample is cut at a half set cylindrical shape, and then you have it's inserted into yeah, you have constraints on all sides, right? I think that's the way.
Yeah, so the sample is only attached at the bone surface. So the attachment does not happen with the cartilage surface. I don't know if that
yes,
makes sense.
but all around the sample, you have it's inputted into a dent, and then your impression pressed from the surface. So I thinking if you have constraint all around the tissue, not only the compression from the cartilage surface, maybe that's the reason why a shrink in all three dimensions. Just along
yeah,
with
so
discussion.
yeah. So again, the cartilage is not in that dent. It is out of that dent.
Okay.
It's only the bone surface that is in the dent.
So the surrounding of the cartilage, and there's nothing hold around it, right?
No, there's nothing holding except the cover slip on the top of the, yeah.
And also,
yeah,
and also you have very high resolution in plane, 0.5 micron, but your slide thickness is 1 micron. So it's not isotropic. If you rendering into 3-dimensional shape of the cell, then your deformation on the depths of the cell, it won't be as accurate as your measurement in plane, right? Because
okay, yeah. So the very first few tests were not done with the resonance scanning. So maybe that's why you saw that. But the was also the correction was made in the Z direction and accounted for the segmentation and the images that were collected.
But your slide, your slide thickness is 1 micron, right? It's not very, eyes height is plane.
Oh,
and you measure the depths change, the changing the depths. It won't be as accurate as in plane change displacement.
Yes. I kind of agree with you for that one. Yeah.
And also, do you count take into account the shrinkage of the full cartilage when you measure the displacement of the cell? If the full cartilage was shrinked, and then maybe your cell position didn't change within that full displacement. I don't know if I can make it clear, but yeah. I think
I understand where you're coming from with respect to that. And I agree with you. I think measuring the local strains around would give us a better picture of what is happening.
Yeah, so I agree. You put it in the future work. If you put the net, then you can in the web, then you can take into account how the whole structure was shrinked, and then your cell is displaced within the whole texture instead of it's moving totally outside of your field of view. And the final question is, you apply the cycle for 100, and then it didn't change anything. Do you think it will stay there forever? I mean, well, will it fatigue if you keep your loading cycles forever? It will fatigue, or the cell will break down, or something really bad thing happens. I don't know.
Well, yeah, that's a question I certainly have asked myself. What if we had a longer loading protocol? Essentially, that's what you're asking, right?
Yeah.
Yeah, I think it would be interesting to observe if at a point we then again begin to, like, would the cell give up its ability of this volume recovery that it's trying to maintain eventually? I don't have an answer, but I guess that's interesting also to see how long it can sort of hold on to this active cell volume regulation process. Yeah.
Okay, thank you.
Thank you.
So Baba, I actually have a question for you as well. So if I'm not wrong, or if I remember correctly, you use 20% of the strain for your testing, like loading cycle. I'm just curious how this 20% of strain is relevant or similar to the knee joint loading that we have in daily activities.
Yeah, so I think it's been previously shown that up to 40% strain loading will sort of be characterized as physiological for the cartilage in the knee. We did start with 20% also kind of because we were trying to set up the whole experimental process, test the protocols, and start from that spot where we could control things to a point that we are comfortable before we can think of slightly increasing the amplitude or increasing the even the frequency of the loading if the device would permit us to do that. Yeah.
And actually the following question that I have, since no one is raising their hands, I'm gonna take this advantage. So if you change your loading strain, let's say 40%, you are increasing the loading strain, then would you expect to see somehow different results in terms of like volume strain and cell strain? Like when you see, like it was recovered after like 10 cycles of the loading in between 10 to 100 cycles. But if you are going way higher loading, would you expect to see somehow it is damaged so it does not recover to the points?
Yeah, I think that's a good question. And with these sorts of tests, I like to think of them as, you know, kind of trying to find what the thresholds are. And really, is there kind of like a sweet spot where the cell behaves in a, you know, is of good behavior, and then all of a sudden it's not of that good behavior anymore. But I can't say for sure what the 40% will look like, whether it's still going to be within the range of that sweet thing or it would have gone beyond that range. But I think that would be interesting to know what the higher strains or the higher frequencies, like the effect they would have on the cells. Most importantly, the recovery during the loading. Yeah.
Thank you. Yeah, I'm just curious if there is like a safety window for the cartilage so that it can bear how much percent of the loading, but it cannot exceed over that.
Yeah.
Thank you. Walter, I see you have a question for Baba.
Yeah, maybe also some follow-up comments, you know, like to your comment, for example, someone, you know, Ryan Madden did once a study where he compressed cartilage to a variety of different static depth. And I think the biggest one was about as much as 70% or so. And what seemed to happen is that the cells kind of deformed more and more and more up to a certain total strain, nominal strain. I think it was about 30 or 40%. But then when you compressed the matrix more, the cell didn't actually deform anymore. So it seemed to be staying at a certain level. So the matrix strain seemed to be quite disconnected, even in this static situation. So it's an interesting type of a thing. The other comment that I wanted to make, Brent brought up some really good things, you know, some things that we don't really quite understand. And just to maybe re-emphasize, in static loading, when we have done static loading, then what happens is exactly what Brent was kind of describing, that the height of the cell decreases. And then along the split line direction, along the collagen fiber direction, you actually get an increase in the length. And parallel and perpendicular to the split line directions, we don't really seem to get much of an expansion at all. And then the overall effect seems to be that you lose volume, but it's exactly, you know, that you get a little bit, that you get quite a bit less height, but then it gets a little bit longer, and then the width, or whatever you want to call it, stays about the same. So it's a very funny type of behavior there, where everything seems to get smaller in all three principal directions. And maybe the last comment that I wanted to make is that what Baba already mentioned, that, you know, cells do have this ability to regulate, I guess, how they look like and how big they want to be. And we've observed that with work with Rami Korhonen when he was a postdoc in the lab, and he put cells, cartilage tissue, and exposed it to different osmotic environments. And then it was quite clear that initially cells under certain environments would be swelling or become smaller, but then they would start self-regulating, and against the osmotic values, against the osmolality, would actually then try to adapt a constant type of a volume. So there seem to be some interesting regulations going on that have not only to do with the mechanics of the thing, but also that the cell somehow can actively react to what's happening. But I have a couple of questions as well. The first one, Baba, how and why did you identify the superficial zone as 100 microns? Because when you looked at your slide that you showed there, then it seemed that for the first, I don't know, 20, 30 or 40 microns that you show, you had very typical cells that looked like superficial zone cells. But then when you go from about 50 to 100 microns, at least from a cell point of view, they seem to have much more the phenotype of a middle zone cell. And so I was wondering, why 100 microns?
Well, first of all, I did mention that we use skeletally immature samples and samples from the young pigs. So really, these you know, zonal differences are not very prominent. But typically, previously, people tend to try to stay between 5 to 10% of the thickness as the superficial zone. But as I had mentioned, the first few layers of the cells, first of all, the density is quite high with the cells pretty much almost close to each other. So there was a little bit of a segmentation issue there, and also even in imaging them as you compress them. But yes, the 100 microns, based on the average thickness of these young pigs, which sometimes can be as much as 2000 microns or 2 millimeters, the 100 microns seem to be that safe space where we could stay in, thinking of the 5%, if we wanted to stay in the 5% tissue thickness.
It just seemed there were nice and flat cells on the surface, and then 100 microns down, they seem to be more roundish. Yeah. Anyway, so the other result that nobody questioned you about, that I think is really curious, is when you showed the results for the middle zone cells, absolutely nothing seems to happen. Can you first tell me how many cells have you analyzed there?
For the middle zone, I think there were about 47 cells.
47, so a good number. Okay, yeah. So therefore the result is probably fairly fairly stable. How do you explain that you don't see anything at all in the middle zone?
So
because
yeah.
Maybe expanding a bit, because GILAC in static loading looked at middle zone cells as well. And when he loaded there, for 20 minutes or however long it was, until he reached some kind of a steady state, he seemed to see a fairly substantial deformation in middle zone cells as well. That didn't seem to be very much different from, well, it was a bit different, but not very much from the superficial.
Yeah.
So do you have any idea why that might be in your experiment?
Well, I don't really know, but then I would think that in the static case with the continuous loading for, I guess, as much as 20 minutes, we, can I say, have that time for the strains, to be transferred to the tissue in terms of the continuous load. But with respect to the cyclic loading, again, I guess we would understand better if we look at the local matrix deformations in the middle zone cells. But even just from the experiment and just going into the middle zones, I don't observe much of the displacement out of the field of view that I encounter as expected with the superficial zone cells.
So if you held your preparation there at 20% nominal strain for 20 or 30 minutes and left it there, would you expect to get the same result as GILAC, or would you expect, because it's a different preparation, you have pig, I think he had dog.
Yeah.
Do you think it might be a difference in preparation, or is it a difference in age because you had immature and he had mature? Or do you think it's a difference in mechanics?
Yeah, well, that was going to be my next point, because I really can't explain it best with the mechanics. But yes, there's also those differences with the young pigs and the older pigs, as well as, well.
So
yeah,
if you did the static loading,
yeah.
If you did the static loading for 20 or 30 minutes and looked at middle zone cells, would you expect to get the results that you have for the dynamic loading, that nothing much happens? Or would you expect GILAC's result that you get a large deformation, a large volume loss?
I think I would expect to see some volume loss with the middle zone cells, be it young pigs or, yeah,
okay,
old pigs, yeah.
Good, thank you.
I see, Scott, you have a question for Baba?
Yeah, Baba, great talk. I really like your, I'm getting feedback here, sorry.
Yeah.
Oh, there's another person watching in the room. Okay, it went away, it seems. I really like your color choices, like the overlay of the cells in the middle zone, especially, like the pastel colors. Look kind of like a Jackson Pollock painting in the end. But anyways, I had a few comments to what Walter and Brent talked about, the cell deforming in all directions simultaneously. That is allowed. Foams do that. So if we imagine a foam going to the bottom of the ocean where it's under constant hydrostatic pressure, it will shrink in all directions. And the cartilage in the cell, especially the cytosol, if you take the fluid away, is very foam-like at steady state. If that's happening at the early stages, I guess it would have to be explained by a big pressurization mismatch between inside and outside. It's a little less simple. It's a little harder to explain just from the perspective of an elastic foam-like material, though. So yeah, it is an interesting point, but we do consistently observe that, so it's something real going on. Also, the comment about the out of plane slice thickness, it's absolutely right. One advantage we do have is that with our segmentation method, we look at what are called the isocontours. So we're not limited to the pixel resolution. We can move that surface, the boundary of the cell, anywhere throughout the volume, right? So it's not as bad in the end as you'd expect. So our resolution isn't limited to the 1 micron jump.
Yeah.
And GILAC showed that in 1995 too. He also used the isocontour approach. So, and I think he showed convergence around 1 micron, or I'd have to look, maybe it was half a micron in slice thickness in his surface area and his volume measurements of the cells. But I do have a question, and it has to do with the superficial zone. We are consistently seeing, and others have seen, that it's so much softer and it deforms so much more, now seen under the dynamic loading, than the rest of the cartilage. And it bothers me because if I were to make a material that I wanted to be able to interface, have low friction, have low wear at the joint when it's interfacing with another material, we want to make that surface as hard as possible. So why, and maybe someone, some other people have comments, why does the cartilage do this? Is it for just moving fluid in and out for waste exchange and nutrient exchange, or is it, what's going on there? Just open question there. I don't know what your thoughts are.
Can I maybe say something? I think one of the advantages of I think you're right when you look at it from a frictional aspect, but one of the things that I always thought was cute about cartilage is that it is a bit soft in the surface zone there, and therefore when you press opposing cartilage surfaces against each other, then the contact area increases almost in a manner that the pressure is kind of maintained. And maybe that's a good thing. And we showed that with Andrea Clark at one point. You press two surfaces together, articular cartilage surface together, and you apply more and more and more force, but the pressure seems to be about the same because you just recruit more and more surface. And if your surface was really hard, then that possibility would go away. So I think it might not only optimize, maybe friction, but it might optimize some other mechanical parameters that might be good for the joint to have. But it's a good thought experiment,
Would there be any other comments on this question or other questions for Baba?
I have one quick other question if nobody else does.
Sure.
What in your system right now is limiting the speed at which we can acquire cell images?
Well, the very first thing I would think about is the resonance scanning has been awesome in terms of the speed. However, the microscope does not allow us to zoom into one or two cells. So then we still have to take like a bunch of cells together. So yeah, it's just that wider space that we just have to work with. But if we were able to, I guess, not zoom into one cell, because then we'll have all that, noise and stuff that comes with the resonance imaging, but at least just move into a few cells at a time, then we definitely could have gone faster.
There be any last question for Baba? Yes, Brent, please go ahead.
Oh, you're muted.
Sorry, you're muted, yes.
There's, if you walk around the HPL, I guess pre-COVID, or probably, a lot of kinesiology departments or faculty departments, you find people that have like these sort of stand-up computer desk kind of things, like not everyone's sitting all the time, and people do a lot of standing, right? And so cartilage does experience, I think, a considerable amount of just sort of continuous creep type of loading. So do you think you're going to maybe look at that kind of like the creep behavior of the cartilage and see if you would see this sort of same thing as you do under dynamic loading, where you have initially, large deformations that then somehow recover it, and it somehow recovers its shape over time? Or Or have you already looked at that?
I actually have not, thought about that. But I guess it's interesting, because all these eventually add to having like the information for the bigger picture in terms of the behavior. Yeah,
Yeah, no, I was just curious. Thanks.
Brent, I mean, the one thing where there's not much that the cartilage community seems to agree upon, but the one thing that everybody pretty much seems to agree upon is that when you apply like a static, long-lasting loading, let's say you stand in front of your computer for 3 hours and don't move very much, that seems to be producing cellular responses that tend to be catabolic. And when you have dynamic loading and you walk or run or whatever, and you have this cyclic loading more that Baba was doing in her study, that seems to really promote anabolic processes. So purely based on that work, standing for long periods of time and not really moving very much is actually not that good, it seems. So standing, if you have one of those standing things, I would suggest that you shift weight all the time a little bit so that it becomes a little bit dynamic.
That's no, that's my feeling as well. I don't know if those things are nearly as healthy as people think they are. But I just have this sense, and I don't have any data to support this, but I have this sense that if you just sort of continuously load cartilage, it's going to act a little bit like a sponge, and it's going to exude fluid, and you're going to, depending on the loading frequency, if you load it fast enough, it's not going to recover its original shape, and it's going to continuously squish down to the point where you're basically cyclically loading it, but it's really just a creep test. It's really almost like no difference as if you would just be continuously holding the load there. And it would be really interesting to see if that's not the case, and to see if actually the cell does something different under those two different, let's call them loading modes.
And just to respond again, sorry, Rob, I'm just going to quickly answer that in one sentence. Biphasic models would predict that, if you did, I don't know how many cycles that Baba does, but it would creep, and it would then at the end reach a steady state type of a situation, like you would if you had a static load. It would just take a little bit longer, but biphasic model would predict that. Sorry, Rob, go ahead.
A statement of my ignorance. The synovial fluid, I presume that there's no pressure inside there and it's sort of a passive bag. But if it was pressure in there, do you actually have another element? If it was under pressure, would the synovial fluid act as a cushion and the synovium itself give you some sort of compression going out sideways? I don't know if anyone's measured the pressure in there. I assume it's probably zero, but
in a joint, it's not. People actually, that's what surgeons are worried about. That's why they don't like to open up joints, because the argument has been that every joint is about under negative pressure, about one negative atmosphere on the negative pressure, and so that the joint surfaces in an intact joint where the capsule has not been breached seems to be under negative pressure, and when you make a hole in there surgically or with arthroscopy, then you breach that. And I've heard from surgeons that they are worried about that. Cy Frank was worried about that. That's why he was very careful before he opened up a joint.
So they were worried it would suck things into the joint?
That you lose that negative pressure that normally exists in a fully intact joint.
But when you get a damaged knee, what do you call that? The Baker's cyst at the back, where the excess synovium has been pushed out the back. Has the pressure gone positive to generate the Baker's cyst? I guess I'm thinking that if I was designing a knee, and I probably wouldn't be very good at it, putting a sac around with pressurized synovial fluid in there gives you another element apart from the cartilage. You've now got a sort of a compressing balloon, but you're saying it doesn't do that. In fact, it's the opposite.
Would there be any other comments on this conversation or any other last question for Baba? Yes, Scott, please go ahead. Sorry, I think you're muted.
I just had one thought that came to me just now. An advantage of a negative pressure could be for capillary exchange with the synovium into the synovial fluid. So you would have a pressure gradient to pull the small molecules that could diffuse. So it would help, pull those nutrients in. Waste out would have to be going against it. So then you would have to rely on the pressurization that results from walking around to help that way, in addition to this passive diffusion. So looking from the mass transport through bulk fluid motion, I think it might have some roles there.
Thank you, Scott. If there is no other question for Baba, I would like to thank Baba again for giving a great talk. I really enjoyed it. Next week, we have an invited speaker from Northwestern University, Dr. Rick Lieber, and the title of his talk is Intraoperative and Laboratory Studies of Human Muscle Contractures. I think it is going to be a really interesting talk, and I would like to thank Dr. Walter Herzog for inviting him. I hope you have a wonderful evening, and I'll see you all next week. Thank you, Baba.
Thanks, Songwan, and thanks, Baba.
Thank you, everyone.
Great job, Baba.
Thank you.